Hello, everyone. This is Kelly Eversole, and I want to welcome you to our webinar this morning. We're going to wait a few more minutes to allow people to join, and then we will get underway. All right, I think we've brought everybody, or most everybody, in. We're still adding some people, so sorry. We'll wait just a few more seconds. All right, so let's get started. Thank you for joining us today to discuss quite a few interesting aspects that's going on with the Genomics platform. The IWGSC is, as most of you know, we're an international consortium, a public-private partnership. We have almost 3,000 members, 2,800 members. We have involvement with over 800 institutes and companies, and of course we have a number of industry partners and sponsors. Sorry. I'm having a little bit of a, there we go. Our vision since we published the reference in 2018 is to enhance breeding through an increased understanding of the molecular basis of traits and their allelic diversity. Our activities this year has been on expanding the IWGSC Arbor Biosciences collaboration. We are still hoping to publish the IWGSC RefSec version 2.0, which includes gap closure of version 1. We are in the process of increasing the annotation, both manual and functional annotation, and we're looking at a version 2 and possibly a version 2.1. It may become before we actually roll that out. We are also beginning the efforts on the wheat diversity project, where we will sequence to high quality at least 8 land races that represent the breadth of wheat diversity, and we continue to try to get pre-publication releases of genome sequences for elite wheat varieties as well as other genomic resources. And the other activity that we had launched in January of this year was our webinar series. And I'm just going to, before we go to the webinar today, I want to give you a kind of an update on the next couple of webinars that are coming up. We have one on the 7th of May that will deal with the Nagoya Protocol and how CABI out of the UK is really dealing with that. And you can register by going to this link. The next one that we will do is a genome-wide analysis of the wheat transcription factor family, and that's going to be done by 2 scientists from the University of College of Dublin, and you can also register there. So today we're very pleased to have Sean Quinn, who's the Chief Technology Officer at Curio Genomics. And he's going to be talking today about extending the Curio Genomics platform for the wheat research community, going from DNA sequence to analysis results. So Sean, I'll turn that over to you.
See, has everyone seen my the primary screen here?
Yes. Okay.
So thank you again for the introduction, and I just want to say thank you for everyone who joined the webinar here and is interested in this topic. I'm very excited to be able to share some of the work that's being done at Curio Genomics in support of the wheat research community, and grateful for the invitation to present to the IWGSC. So just to kind of get started, I hope everybody's seeing the slides as I switch here. I just want to briefly just mention kind of what I plan on going over here at the beginning. So we're going to share a little bit about what Curio is for those that may not be familiar with Curio Genomics. And then we're going to be spending most of the time going through several different crop and wheat research examples in the platform. That's both DNA-seq as well as RNA-seq data, covering everything from read mapping, alignment and analysis and visualization of your read data, as well as through different kind of interpretive analyses of things like variant analysis, coverage analysis, as well as expression and differential expression analysis of some RNA-seq data. And we're going to be using several different data examples that have been worked on with other collaborators of ours within the IWGSC, and I'll be sharing some of that as we go. And then at the end, I have a few thoughts to share about what's coming ahead for us and other things we're working on before we go into hopefully some time for some Q&A. So with that, I'll dig in. A little bit about what Curio is. So Curio is a big data management and genomic analysis platform, and it's fully web-based, and so it's a little bit different than other tools in that it really provides a way to both analyze and interpret the data right within the platform, which makes it right within a web interface, which makes it very compelling for collaborations both within organizations as well as across several organizations. It supports, it really serves both the bioinformaticians that are trying to do bioinformatics data processing of their samples, as well as trying to support the biologists and the researchers that are trying to ask interpretive analysis questions of the data. So it helps with the bioinformatics data processing, job management and running of analyses, but then also allowing you to actually interact with the data and start asking interpretive questions of the data that a biologist might be interested in. And one of the ways it does that is it's actually built on a unique data processing clustering technology that allows it to actually take any analysis job that you run within the platform and actually break it down into smaller components and spread those components out across a large cluster of computers, which then work on the problem in parallel and then store all the data in a very unique way in a real-time database so that you can then ask questions of the data essentially instantaneously, because you don't have to wait on files to move around or tools to run. Everything's happening sort of at your fingertips by being able to ask questions of this real-time database instantaneously, as well as running any analysis jobs that need to run in parallel across many computers in a cluster. It's really designed from the beginning for extensibility, and so we're constantly working with researchers and collaborators to add new types of analyses, new types of data. As everybody I'm sure on this webinar knows, this space is moving quickly and it will never be done. And so part of the design of the platform and the way it's architected is to really allow us to continue to evolve it quickly and efficiently, rather than having to wait on major releases for new functionality. We're constantly evolving and extending the platform, both in ways that are proprietary to certain institutions, as well as ways that are shared and available to everyone using the platform. And what's relevant for this webinar is one of the things we did last year was we added several different capabilities to support crop research and agricultural research in general. And particularly relevant for this webinar is the addition of both hexaploid wheat and tetraploid wheat based analysis of both DNA-seq and RNA-seq data. So with that, I'm going to dig into talking about a few different types of analyses that you can do within the platform. The first is to say a few words about read mapping and how alignments are handled within the platform. So one of the things you can do within Curio is you can actually just select from the IWGSC reference assembly as what you want to align to. And as part of that, you can choose from different industry standard algorithms for how you want to take advantage of the reference assembly to do your read mapping and alignment phase. As part of that, we've already have built into the system all of the necessary indexes and data and annotations that go along with each of these choices. And so as soon as you hit the button, your data will start aligning instantaneously because everything is sort of ready to go. One of the unique things about the way Curio handles the read mapping and alignment phase of an analysis is, instead of normally you would have to stitch together lots of different tools in a bioinformatics pipeline to do things like read trimming or quality or downsampling or so on, where you would stitch together a series of tools that would then create subsequent FASTQ files, which would then go into the next stage of the pipeline and so on. Curio actually handles things very differently in that it simply allows you to sort of, through the user interface, just tune those options all in the same step. And so when you start an alignment within Curio, you can do things like quality trimming, downsampling, UMI processing, adjusting your scoring parameters and so on, all at the same time. And what Curio does is it spreads that analysis job out across a cluster and does all of that work sort of simultaneously, so that it doesn't have as many intermediate files that it's processing. It's basically just pushing the data through all in parallel and running all these bioinformatics steps all at the same time, and allowing you to experiment with those options so you don't have to wait on jobs to finish. You can kick off a job and see how it's behaving within minutes instead of having to wait for an entire pipeline to complete. So you can quickly experiment and see how different settings are affecting your analysis. Once you have the data mapped to a genome, you can then visualize it. And so here is a screenshot of what the alignment visualizer looks like within Curio. You can see up here at the kind of the top left, gives you a quick overview of how your reads mapped across the entire genome. Everything from chromosome 1A through chromosome 7D, as in this case, we're looking at a hexaploid wheat example that was aligned to the Chinese spring wheat reference genome. Down at the bottom, you can then start jumping around and looking at your read data and your mismatches and your alignments sort of instantaneously, as you can browse the genome and jump to different positions of the genome to see how your alignment data is looking, including things like your mismatches. So you can see here where all these little colors are highlighting different mismatches of the read data from this sample when compared to the IWGSC reference genome. If we want to toggle those options, we simply adjust one of these options up here at the top of the screen, and Curio instantly reprocesses all the data for us, because again, it's all in a big data database, so we could just run those queries with different parameters, and then we can see how those different quality settings are affecting things like read coverage, the mismatches, as well as how things like PCR duplicates and whatnot are affecting this area of the genome. But then beyond even the reference assembly, we can start taking a look at the great transcripts and functional annotations from the IWGSC inline. So here up at the top of the screen, you see which annotation sets we have selected right now. Right now we have the high confidence genes selected. And we're then down here at the bottom of the screen seeing the transcript information from that genome alongside our actual sample read data, as well as you can see this coverage chart of the read data at this area of the genome. If we hover over that, we can see things like where the exons are, where the coding regions of those exons are. You can see the intronic regions here are visualized. And then beyond even the transcript annotations, we also have the functional annotations sort of synchronized with all those transcript annotations. So you can see here on the right, we want to search for a keyword, in this case, the keyword glutenin. And we want to find all the different transcripts that have been annotated with that functional annotation according to the IWGSC annotation set. If we hit enter, Curio then instantly jumps and visualizes for us that area of the genome. In this case, it's a gene near the beginning of chromosome 1A, and we can see the different SNP calls and whatnot starting to come through, again, with all the filters and whatnot applied on the fly. Similarly, because this is all, again, in a database, we can just simply ask Curio to give us the data. And so if we want to just download, for example, the consensus sequence, Curio will on the fly collapse all of the read data and develop a consensus sequence, as well as the genotyping of all those calls that it could find in that area of the sample. Or what's being visualized here is we just want to get access to the IWGSC reference genome for this area of the genome. Again, it's all in the database, so we hit this button and Curio instantly builds for us a FASTA file that represents this area of the genome so that we can grab it and use it if we need to look at that raw genetic sequence for any area of the genome. And then another neat thing about what Curio does to sort of enable, to try to help enable collaboration is anything you're looking at in Curio on really any of the screens, you can mark through annotated comments. And so you can make a comment and mark that you want Curio to keep track of whatever you were looking at. So in this case, we were looking at one area of the genome that has a couple of variants of interest. Maybe this deletion that appears to be found here might be of interest. You can record that. And then whenever you want, you can go back and remember what you were looking at by just simply clicking on those annotated comments within the samples, or share those comments with other people on your team or other research institutions so they can also access that same annotated information that you've added to your samples. Once you have data aligned in Curio, you can then analyze it in different ways. So here's a screenshot of how you kick off a variant analysis where you might want to look for variants across one or more samples. In this case, you can see down at the bottom of the screen here, kind of grayed out behind this dialog, there's about 1.5 billion reads that we're about to ask Curio to analyze in parallel across these half a dozen different samples. We can then tune these options in a user interface that makes it fairly easy to understand for either a biologist or a bioinformatician to understand how they want to potentially tune the way these variant calls are going to run. So you don't have to read through a lot of command line tools. You just look at the user interface. Little help icons are available if you want information behind what any of these options mean. And then you can tune things. So for example, if you wanted the variant analysis to be higher sensitivity, you could tune the minimum quality slider, or you could decrease the minimum read coverage, or adjust the frequency at which you are going to require evidence of a variant to be present across your reads in order to call it as a variant. And so you can sort of see here how you can just kind of easily just tune those options to adjust your analyses, click the button, then in parallel, Curio will often within minutes, run a variant analysis for you across these samples in parallel. And if you want to tune the options a different way and run another variant analysis, just simply adjust the sliders and hit the button again, and you'll get many different analyses run with different settings on your same sample set. One of the nice things about doing your work in a platform like this is it's also keeping track of everything for you. So for example, in this case, I clicked on one of the files, one of the alignment mapping files here, and it's showing me where that file came from. It came from this FASTQ file pair. It also shows me the different analyses that have already been performed on this particular sample. So there's 3 different variant analyses that have been run, and then I can sort of drill in and then look at any one of those. I can then go a step further, and if I want to know even the specific parameters and the versions of tools that were run to build those analysis results, you can see it's tracking for me here all of the different parameters that I chose, as well as the versions of the tools that were run, so that we can know the provenance of any analysis or any result that we got in the platform is all sort of being tracked for you, so you don't have to remember all the settings you chose to get to any one result. Once you have the variant analysis kicked off, Curio will then build for you a screen that looks, a report that looks like this, where again, this is a hexaploid wheat example. And so we're seeing where all the variants are across the different chromosomes from 1A through 7D. And we're then beginning to visualize down here at the bottom of the screen what those variants are. And so they're being color-coded in different ways. This data is actually from some interesting research work that the John Innes Center is doing, where they actually had a sample that was a series of wild type samples that they sequenced, which had some phenotypical qualities that they were interested in. And then they actually irradiated the seeds with gamma radiation of those wild type samples, and then through several generations later grew those plants, had those child descendant plants sequenced, and then they wanted to look for areas of interest of mutation that were present or not present in the descendant organisms that were not present in the wild type, so they could sort of understand when the phenotypical benefit of the wild type sample was either found or not found in the child descendant species, or excuse me, descendant organisms. So then they could identify what are these variants, what are the areas of the genome, what mutations are actually affecting the phenotypical benefits or lack thereof that we're seeing in these wild type samples. So here in Curio now, we're visualizing those 3 samples side by side. Here, the control sample is shown at the top, the wild type, which was run with higher sensitivity, which is why you see more variants present, because we wanted all potential mutations that we could potentially find in the wild type to be part of our analysis. We then want to ask Curio to give us to do an analysis again on the fly, show us just the variants that are present in those descendant organisms that were not present in the wild type. So now, because we had a very highly sensitive variant call set in our wild type, we've narrowed this down to only variants that are likely uniquely present in areas of the genome that have mutations that are likely present in those descendant organisms that were not present in the wild type. We can then start leveraging the annotation sets from the IWGSC to ask even deeper questions. So one of the great things about the IWGSC annotations is it defines where all the genes and transcripts and coding regions are. And so right now we have the high confidence genes selected up here at the top. And we could just toggle that on the fly if we wanted to use, for example, the low confidence genes. Over here, then we've said, hey, Curio, show me just the variants, just the mutations that are present within coding regions of the genome according to that annotation set. And now we've narrowed it down to a smaller set. Now we've narrowed it down from, hundreds of thousands to only 74 variants of interest, at least within this one particular area of the genome. We are then going to go a step further, and Curio will on the fly calculate, because it knows where those coding regions are from the annotation sets, it can actually calculate the amino acid shifts associated with each one of these variants that we find within the samples. And as it calculates those, it can then build for us a color-coded chart of what those, where those variants are. So here we've narrowed it down, and we're looking at just the variants within the samples that were not present in the wild type, because we've turned on this semantic analysis, that are only considered non-synonymous as the predicted biological consequence of each of those variants that were detected in the samples. And then we can go deeper. We can go even deeper if we want. So we can toggle the visualization and color code our variants in different ways. And if we want to see what's behind even one of those variant calls, we can hover over it, see the gene and the transcript information from the annotations, as well as click on this view alignments link, which would then take us directly into the raw read data, the visualization we saw earlier, to see the individual read and sequence data that was behind each one of these variant calls. There are many different types of analyses you can do within Curio. I'll briefly just mention another one here is of DNA-seq data. Another one is coverage analysis, where in this case, we want to measure the coverage of our libraries or our kits and see how well they're doing across the genome. In this case, we ask Curio to measure for us the coverage of the exome of the Chinese spring wheat genome according to the high confidence annotation sets from the IWGSC. It did that analysis for us. It's giving us some metrics and some visualizations of how the data coverage looks across those areas of the genome. And we can, again, compare data. So here we want to compare, how do different alignment mapping algorithms affect our coverage results of the sample. So this is the same sample run with different alignment mapping algorithms, and we're visualizing here how that affects our coverage metrics across the genome, both for on and off target here in this pie chart, as well as per chromosome in this column chart over here across those 3 different samples we have selected. And if we want, we can go even deeper and look at individual genes, transcripts, exons. So you can see here a visualization where we've jumped to a particular transcript and we're trying to get a visualization for how do the different exons in that transcript actually, how do they seem to be affected by the different alignment and mapping algorithms we chose. We have 4 different ways we map the data, and we can sort of see in this case, Bowtie in local mode seems to do a better job at coverage for this particular gene that we're looking at from a coverage analysis perspective. So those were a couple of things about DNA-seq. I just want to mention a couple things about RNA-seq as well. So one of the things that Curio does that is really cool is that it can leverage the IWGSC annotation set to actually build on the fly a transcriptome for you. So if you select here in an RNA-seq analysis the alignment mapping algorithm of top hat, you can then select what annotation set you want it to use. You can either use one of our standard built-in annotation sets, which you can see here are showing up in this dropdown, or you can upload your own annotations, your own either bed files or GTF files to define how you want to define the transcriptome. Curio will then on the fly build essentially a different reference from the IWGSC reference assembly you have selected at the top here, and it will build a transcriptome of that for, based on all the exons and transcripts defined in that area of the genome. And it will then build for you, it'll map that data to the genome. So then when you go and you actually look at that RNA-seq data in the alignment visualizer, you can see right away how your data is then aligning much more nicely, your RNA-seq data is aligning much more nicely as you might expect it to the genome. If we had run this same analysis and just use a standard mapping algorithm to map this same data to the reference genome instead of the transcriptome, we would have had much lower coverage data and much worse alignments because a lot of these reads either wouldn't have aligned at all or they would have had either soft clip or mismatch regions in this area of the genome. So you get much better coverage of your data, but also you get much better alignments because you can see how the alignment mapping algorithm is both allowing us to process as well as visualize how each of these individual reads is actually spanning over the intronic regions and aligning nicely to the exons, which again, we're visualizing here on the top of the screen, these exons coming from the annotation set provided by the IWGSC. Once you have your data now visualized and aligned to the transcriptome, your RNA-seq data aligned to the transcriptome, you can then start trying to correlate that with the IWGSC information. So here we're showing where the exons and coding regions are in this area of the genome. But now we want to ask a deeper question, which is, what do the expression levels of the genes look like across these different RNA-seq data? So here, you can see here down on the bottom of the screen, we've got another 1.5 billion reads or so that we want. There's actually more than that. There's a couple billion that are sort of down below that we want Curio to analyze across 8 different samples. And this is RNA-seq data that we've aligned to the transcriptome. And so we want to ask Curio to calculate for us the expression level of the different features of these samples, again, based on the annotation set that we've selected here from the IWGSC. Or again, you can upload your own feature sets if you have your own definition of where you'd like to measure the features within the genome. And then you can ask Curio to measure the expression level of the features different ways. So you can, because we know where the genes and transcripts and exons are in this high quality annotation set from the IWGSC, Curio knows that it can measure the gene expression, which is what researchers are looking for. But you can even go deeper and measure the expression level of the individual transcripts or even the individual exons across the genome, again, according to the annotation set you have selected. You can then start a differential expression analysis on that data where you basically select all the samples you're interested in, put them in groups, tune the different algorithm options that you want Curio to use when it's going to run the expression, in this case, the differential expression analysis. So here you can see we're tuning a couple different industry standard algorithms that Curio is going to use when it's calculating the differential expression. In this case, DESeq2 and EdgeR, you can tune the options that are going to affect the way those algorithms calculate the differentially expressed features. And then Curio builds for you a report that looks like this, where you can then start looking at and visualizing the expression levels of the different features that you've asked Curio to measure, again, using that transcriptome-based alignment, leveraging the IWGSC reference and the corresponding annotation sets. So this is kind of another interesting study that was actually done back in 2015, I believe, where a research group was trying to measure and determine how gene expression changed across different wheat organisms as they were exposed to different conditions. So they actually sequenced a control group of wheat, and then they also sequenced a group of wheat that was exposed to drought stress conditions, also a group that was exposed to heat stress conditions, and then a fourth group that was exposed to both drought and heat stress conditions simultaneously. And then we drop that sequence data into Curio, ask Curio to measure the expression, and we're beginning to now get some visualization of what that data looks like. So here we have just a quick visualization of what the feature count expression levels look like across those different groups. We can then go a step further and ask Curio to show us, how did our data cluster? Is it clustering like we'd expect? This is a key part of any differential expression analysis, is you need to know, does my data cluster as I'd expected? Because if it doesn't, I really can't conclude anything useful from a differential expression. Well, in this case, the data clusters quite nicely. So you can see over here on the right, you can see how the different samples are clustering naturally on their own without any bias across those 4 different groups that we expected. That's based on the options we have chosen up here at the top of the screen. So if we adjust one of these options, it'll rebuild these clustering matrices for us on the fly. Similarly, a key part of any differential expression analysis is seeing how did the groups cluster according to a principal component analysis. Again, in this case, you can see the data looks great. These 4 different groups are clustering very nicely and independent of each other between the control and then the 3 groups that were exposed to the different stress conditions. We then want to go a step further and ask Curio to tell us, again, according to those annotated data from the IWGSC, how do these samples, which genes are the most highly differentially expressed? In this case, we're looking at those that are strongest either up or down regulated. And so you can see here, we're seeing both genes that were more highly expressed in some samples versus the others, as well as those were more lowly expressed in some samples versus the other. And you can already see just from this heat map visualization, some interesting results of, okay, we've got some genes here that are clearly much more highly expressed in the organisms that were exposed to drought. But those that were exposed to heat, we had a completely different set of genes that were more highly expressed, and those that were exposed to both drought and heat maintain those same, those same genes were continue their expression, but those that were used to be expressed from the drought conditions, for whatever reason, were not expressed, at least not at the same intensity in the samples that were exposed to both drought and heat stress conditions. So you can, again, just from these options, you can sort of begin asking these interpretive questions to your data through these visualizations. And again, everything is sort of on the fly where you can adjust things. So this slide, we were looking at the DESeq results according to these settings that we've selected over here on the right. If we want to tune those options and look at the data differently, we just simply click a button. The data is all in a database, so it kind of rebuilds those analyses on the fly for us. And now we're looking at, and the EdgeR algorithm instead of the DESeq algorithm, which, the data, it's still similar genes expressed differently, but a few are different here based on how the EdgeR algorithm is calculating the differentially expressed features as opposed to the DE-seq algorithm, again, under these certain p-values and settings that we've chosen up here at the top of the screen. So that's differential expression. Just to say a word about tetraploid wheat as well. So everything I showed up to this point was leveraging the IWGSC Chinese spring wheat reference and related annotations. The IDWGSC also has a nice reference available, as well as annotations available for tetraploid wheat, for durum wheat. And so here you can see a similar analysis where we've mapped the data to the tetraploid wheat durum reference. And now we've got a chromosome 1A through 7B of chromosomes available that we can visualize the alignments across these samples, get an idea of how our reads are mapping, and the quality of that data is sort of coming into different areas of the genome. Look at these annotation transcript data, again, from the IWGSC, and take advantage of that as we visualize the data. We can then, this was then a study, some work that was done by Arbor Biosciences, where they wanted to see how some different samples aligned and mapped, and how they could basically see how the density of variants were found in these different durum wheat samples when by leveraging the SVEVO wheat reference assembly from the IDWGSC. So again, they align the data, ask Curio to call variants, and you can already see just from this visualization down here at the bottom of the screen, across these 8 different samples, we've got a lot more variants in this 7th sample and a lot less in this primary analysis file up here at the top, and a range of different density of variants for the different samples in between. Again, we're leveraging the IWGSC's annotation set here, so we can start doing things like filtering. So this visualization has already been filtered to only show variants across these 8 different samples that are within coding regions, according to that reference assembly. And they've been color coded and highlighted in a certain way. And then if we want to go a step further and say, hey, only show me those, I'd like the data to be visualized in a different way. Instead of looking at them by their genome type or their genotype, I'd instead like to look at them based on their predicted consequence. So now here we're color coding from sort of green to red, a spectrum of the different intensities of amino acid shifts that would cause a predicted biological consequence of each one of these variants. And let's go a step further then and ask Curio to just show us those that are going to have a predicted biological consequence of a stop gain in one of those codons. And so now we're narrowing it down, and we've got a much smaller set of mutations that we're looking at based on, by leveraging both the reference and the annotations, along with, in this case, several billion reads that we're trying to analyze simultaneously to kind of get these different variant calls that we are trying to analyze and ask questions of across this cohort of samples. With that being said, just a few things about what's coming. So as Kelly shared here in the introduction, the IWGSC already released a 2.0 reference assembly and is in process on working on a new set of annotations that will go along with that reference assembly, either 2.0 or 2.1. And so we're anticipating that. And as soon as that's available, we plan on adding that into Curio so that you can not only just use the reference, but also use all the benefit from all of the power that those annotations provide to be able to ask, questions of exons and transcripts of this data, not simply just information we have from the reference sequence. So we're excited about when that becomes available. We plan on adding that in as just another option that researchers can use when they're running analyses in the platform. We are also in process and working on other species that we can add in. Certainly some other wheat references and annotations are beginning to become available that we can use, as well as other related species like barley and rice and so on that we could potentially add into the platform as we collaborate with different researchers that would like to leverage the platform for different types of species and genomic analyses. And as part of that, one of the things we're working on is the ability for researchers to leverage their own custom reference assemblies, either those that have been published to the public or those that you've built on your own through de novo or other ways. So you can, we're working on a feature so you can essentially upload those reference assemblies into the platform, and then Curio will on the fly build all the indexes and get all that data in the big data store in the same way that it can for all of the standard reference assemblies that we already support out of the box. And we're always looking for other research institutions, other collaborators that are looking to extend the types of analyses that they can do in the platform. As I said at the beginning, the platform is really built for extension, and so we are constantly looking for collaborators who want to ask the next question, who want the platform to do more for them. So we're working on already with ideas with a couple of collaborators that are going to be adding some new analyses types and new data types into the platform, and certainly are looking for others that would like to collaborate with us in that way on really extending the ability to do crop research specific analysis types directly within the platform. Before we go to Q&A, I just want to say a special word of thanks to collaborators. Folks at Arbor Biosciences have been key collaborators for us. The John Innes Center, we started collaborating with last year, as well as folks at INRA and the University of Adelaide have been great collaborators, both in some of the data that you saw in this presentation, as well as just helping us as we've sort of stepped into the agricultural genomic research community with the platform. And so very grateful for the collaborators at these institutions that have been a great help to us. We're very excited to continue working with them and other collaborators as we step deeper in to crop research analysis solutions. And of course, I want to pause here and say, without the efforts of the IWGSC and the IDWGSC, none of this would be possible. The reference assembly and these annotation sets that the consortium is developing are incredibly high quality and super valuable, and without those, we wouldn't be able to do any of this. And so it really serves as the foundation for what we're trying to do with the platform, is really trying to enable, great research, great analysis, more efficient results, higher collaboration by really leveraging these great data sets that are coming out of these consortiums. And for those that are interested, I've included here the details on the study where we got the FASTQ data for the differential expression analysis that we showed, which had to do with looking at expression levels in wheat organisms that were exposed to heat and drought conditions. So with that, I'm going to pause, and I guess I think I'll pause here and turn it back to Kelly, and we can move into a time of Q&A as we have time.
Great. Thank you very much, Sean. A great presentation and overview of everything you guys have been doing. It's really impressive what you have been able to do with the reference sequence from both the hexaploid wheat as well as the tetraploid. If you have any questions that you'd like to have answered, I think we do have one. Yes, but it's actually any idea when the IWGSC version 2 annotation will be released? Sean asked me that this morning. We don't really have a firm date at this point, but we do hope that it will be within the next few months. We are busy working on that, and under the leadership of the group at INRA, and we do hope that we'll have it out as soon as possible. There are some areas that we've tried to make sure what we're putting back out is correct, and so that effort is underway between INRA and the group in UC Davis, and also Jane Rogers is working with us in a leadership role to try to pull all of that together. So that is underway, and we hope to have an announcement as to timing in the next couple of months. Okay, let me get to another one. Yeah, I think that's, if you have any questions, you can put them on send them in through the platform. All right, here we go. We have, first of all, a comment. Thank you for the interesting talk, and the Curio platform seems promising. So the question is, from a computer hardware requirement, what does it take in terms of RAM and storage to run the analyses?
So good question. So one of the things that's unique about the Curio platform is we actually manage it for you. And so it is a cloud environment. So everything you saw here, although it may not have been obvious, it actually wasn't a desktop application. It was all in a web browser. So all of these visualizations, all the different dialogues that we were showing, were all actually just running in your web browser. And so there's actually very small memory or computational requirements to run it, because we're really only depending on the web browser to interact with the platform. Behind the scenes, we're running the data then on a cluster, and we're running on a large cluster of machines that are basically tailored for the different pieces. So we have one set of machines that are running the database, the big data database, which is a cluster of data nodes that are handling making all this data instantly available. There's a different cluster of machines that actually do things like the alignment jobs and the variant analysis and so on. And each of those machines has different requirements. So most of the people working with us simply either upload their data or import it through an agent into the platform, and then you just simply use a web browser to interact with it. And so there's really no hardware requirements. You can literally run this entire interface even on your phone. For those that are interested, though, we also do offer an on-premise solution or a private cloud solution. And if you're interested in that, basically running your own instance of the entire platform, not just the user interface, or if you don't want to leverage our cloud version for whatever reason, we do have the ability to do that. And I can get you more information if you want to reach out about essentially the hardware requirements for those that are interested in running this, the entire infrastructure stack in your own environment. But again, most people don't need to. You just simply upload the data to us either through a web browser, or we have an agent that will handle the uploads for you. And then all you need is a web browser, and even literally on your phone, you can run and interact with all the same kind of things we were showing here during this webinar.
Great. The next question, and you mentioned it in the previous one, is there a limit to upload the sequence data? Is there a limit to the amount of data?
No, there's no limit. So, we have people using this data, we have some people uploading small samples that might only have a couple million reads to people literally uploading, samples that have hundreds of millions, potentially billions of reads. And that's totally fine. There's no limit on file size, no limit on the number of reads. You simply upload them, and Curio will break all that data down into smaller parts and process it all in parallel. So there are no file limits, no size limits. Another interesting thing to say about this is Curio actually has a unique way it actually even does things like the representation of the chromosomes themselves, as well as the way variants are called. So you'll notice, for those that have done work with these references, they're really big because wheat is a massive genome. The chromosomes are much larger than the human genome. And so one of the problems that comes in with a lot of traditional tools is that you have to break the reference down by splitting the chromosomes in half and whatnot. You don't have that problem with Curio, because we actually have a different way we store and represent the data, and the way we bin the data and whatnot. And so we don't have the same limitations that a lot of traditional tools have, because we basically have worked around that by having our own strategies for the way we bin and organize the data. So yeah, no file size limits, and you're also not impacted by the fact that these chromosomes are larger in length than many traditional tools can handle.
Great. How frequently is the software versions updated?
We roughly have a quarterly schedule, and so the interface itself that you see is roughly being updated with new features on a quarterly basis, is sort of the pace at which we're adding features. We often have small sub-releases in between quarters, but roughly about 4 times a year, you can see new features and new capabilities get added to the platform.
Great.
And again, that's basically seamless to you, right? Because it's a web interface. you just simply log in tomorrow and you have a better version of the software available to you instantaneously. You don't have to worry about installing anything. So that's one of the nice things about managing it through a cloud environment.
Right. So do you do any additional analyses such as gene ontology, term enhancement?
Not right now, but that is one of the things that has come up in how we can basically go further. We already do a lot of primary, secondary, and some tertiary analysis, but when you get into things like gene ontologies and whatnot, that's even deeper in. And so those are some things we would like to collaborate on, but right now we don't have those kind of analyses built into the platform, but are interested in doing that if there are research institutions that would be open to collaborating with us on that.
All right, great. So how is the data privacy handled if you are a private company? Can you keep certain things, or not even just a private company? If anybody wanted to keep certain things private, there's no problem with that, correct?
That's right. So we take our sort of the protection of the data very seriously. And so and I didn't really show this in the screenshots, but one of the things you can do in the platform is there's very tight controls over who can see what. And so when you set up an organization or an account within Curio, the only people that can see that data are people that you give access to your organization. So I was looking at one organization here when I ran through this demo. But as I log in, I only have access, even me as the CTO, I only have access to the data that is within organizations that I have access to. There are many other organizations within Curio and I don't have access to that data. So you can only see what you have access to based on the organizations that you're a part of. In addition to that though, so all that data is sort of safe and protected and it's only available to those that log in with the correct accounts. But in addition to that, you can also collaborate and share with any of the data. So in addition to making comments, you can just choose to share either an entire project or even individual analyses with anybody you want. And so when you do that, you can basically choose what rights you want to give them. Do you want to give them essentially read-only access to the data so they can look at it and download it? Or do you even want to invite them in as a collaborator to one of your projects? And so you have that ability, kind of like you would in an environment like Google Docs, where you can just sort of set up who you want to have access to your different projects. The same is true here. You don't have to limit yourself to only those that are within your organization.
Great, thank you very much. How much storage is provided in a standard Curio license, or does it matter?
So we have different licenses scales. Most of I should mention that we have a great collaboration with Arbor Biosciences. And so if you are using kits from or myReads or other services from Arbor Biosciences, we're actually developing or strengthening our partnership this year so that as you get those either kits or services from Arbor Biosciences, you'll have access to essentially a read-only version of some of the data directly within Curio. For those that then want to go deeper, our base license starts with essentially around a size of about 100 samples. And so it includes enough storage and data processing to include at least 100 samples. And then we, as you need more, there's just essentially an add-on to be able to buy more samples as you need them. So we're generally giving you at least 100 samples with a base license, enough storage and data processing to support that. And of course, as depending on what you need, we can scale that license appropriately.
Great. Are there, other than the modules or the things that you've added and you're looking ahead, are there additional modules that you're thinking about adding over the next year or so?
Yes, I think one of the key, there's a couple that I'd mentioned. One is on the variant side. We're actually already collaborating on a way to look at the way to do, to get useful information from the mutation and variant calling that you can get out of this, because we have a lot of researchers looking for ways they can look at genotype information on lots of samples, these large cohorts, and be able to find regions and sequences and genes of interest that they can then study deeper. And so that's one of the things we're really beefing up in adding even more to our variant and mutation analysis capabilities across cohorts of samples. The other one I'd mention is that we have a growing interest in people being able to correlate both genotype information with phenotype information. And so we don't have this very well nailed down yet, but we're basically starting to work with folks that are interested in how can they use phenotypical data that they have on these samples and correlate that with the genotype data that we have that the platform is able to call for us so well. You know, how can we start better finding genotypes and areas of interest by correlating and allowing you to keep your phenotypical data directly in the platform alongside your samples, so you can correlate that with the genotype data that's already well handled within the database.
All right, great. The next question is, do you plan to add wild immer wheat, the genome Zavetam?
Yes, that has already come up, and I think that's one of the species we're looking at. And frankly, just to be honest here and transparent, we're kind of right now in the process of analyzing which other species we want to just add in so that they're built in and available to researchers. So I would really encourage you to either send me a note or reach out to somebody through the website on the Curio team, and we would love to know which species and references and annotation sets you could benefit from. And because we're sort of doing an analysis right now to identify which of those we could add in. And we certainly are biased towards those that have really rich and capable annotation sets, because that really showcases the power of the platform. But even those that don't have strong annotation sets, we would be interested in, depending on what you need. So please reach out if you have a need for certain species or references, because that right now would be a great time to influence our product team as we establish those priorities.
Well, someone just commented that in fact Barley would be great, but you mentioned that in your presentation that you are looking at potentially adding Barley to the platform.
Yes, yeah, Barley is on the short list as one that's already been highlighted, but there are others I'm sure that will be popping in this year as we get requests from different folks.
All right, I have one last question that's come in from our participants. How long does it take to go from aligning FASTQ to SNP variant calling?
It depends a little bit on your sample size, but say if you have, say, 50 million reads, you can get from FASTQ data to variant calls within less than an hour. And we're actually in the process of actually enhancing our infrastructure and the way we do our clustering, our data processing, to make that even faster. But often it's, for smaller samples, it can be minutes, but no more than an hour or two, even for large samples. And again, that happens all in parallel. And so even if you have a large number of samples, all that data processing is happening in parallel across the cluster.
Great. Well, thank you very much. You mentioned earlier that you're open to collaboration, so I would encourage all the participants who are interested in that collaboration to reach out to Sean. And your email is actually in your presentation, I believe. So, and everyone should be able to download that. But I want to thank you very much, Sean, for an excellent presentation today. And this presentation will be made available on the IWGSC YouTube channel. And so you can play it at any point in time. You can also share it. And we do encourage everyone to let us know if there are areas that you'd like for us to do additional webinars. As I mentioned earlier, we do have a couple that are set up for May, and we look forward to seeing most of you again. So thank you very much, Sean. We appreciate your time this morning and look forward to seeing you when we get through this lockdowns that we're all in at the moment. So thank you.
Indeed. Be safe, everyone, and thanks again for your time. Have a good day.